Hello and welcome to this Fritsma Factor module, Cell-Based Coagulation. I'm George Fritsma, your narrator, and our module series in the Fritsma Factor are sponsored by Precision Biologic Incorporated of Dartmouth, Nova Scotia. PBI provides quality frozen hemostasis materials for the in vitro diagnostics industry. This module will take approximately 30 minutes to complete. Cell-based coagulation is a detailed refinement of the traditional plasma-based coagulation model. Cell-based coagulation was first described by Hoffman and Monroe in 1999 and consists of 3 cell-bound phases: initiation, amplification, and propagation. We study the phases as though they are discrete, but this is purely an academic contrivance, as the 3 phases are intimately intertwined. Indeed, some researchers prefer to combine amplification and propagation as though they were a single phase. Initiation could be thought of as an idling phase with no measurable clinical outcome. Tissue factor that is constitutive on fibroblasts and smooth muscle cells or is induced on monocytes or endothelial cells may become exposed and decrypted. It then cleaves and activates factor 7, forming 7A, and then forming the tissue factor 7A complex. The tissue factor 7A complex then cleaves factor 10, giving you 10A, which cleaves, activates, and binds factor 5 to form 5A. The 10A, 5A complex cleaves and activates prothrombin to form thrombin. Tissue factor 7A also cleaves factor 9 to form 9A, but with no factor 8 around, the 9A cannot function. All the free initiation proteases are bound and inactivated by tissue factor pathway inhibitor, Z protein inhibition complex, or antithrombin. Initiation occurs away from an injury site and outside of vessels, often within the lymphatic system. This image illustrates the verbal information from the previous slide. Tissue factor is exposed and decrypted. It cleaves and activates factor 7, forming the tissue factor 7A complex. Tissue factor 7A then cleaves factor 10 to form 10A, which cleaves, activates, and binds 5 to form 5A. The 10A 5A complex cleaves and activates prothrombin to form activated prothrombin or thrombin. Tissue factor 7A also cleaves factor 9 to form 9A, but 9A cannot function. All the free initiation proteases are bound and inactivated by TFPI, ZPI, or antithrombin. While we think only of tissue factor exposure in injury, we've learned that tissue factor has modest avidity for factor 7 until it is decrypted. 4 proposed mechanisms are shown on this slide. The first parallels a plasma membrane function common to platelet activation, in which lipid transport enzymes nicknamed flipase, flopase, and scramblase rearrange the membrane so that the negatively charged phosphatidylserine moves from the inner to the outer layer, to orient plasma factor 7 to a joined tissue factor and become activated. Mechanisms 2 through 4 propose changes in the tissue factor molecule, including oxidation, dimerization, or fusion with phosphatidylserine-rich plasma membranes. Tissue factor is also induced on monocytes and endothelial cells, primarily by gram-negative organism lipopolysaccharides and also by viral ligands. Tissue factor is also found on the surface of polymorphonuclear neutrophils and platelets, perhaps absorbed from monocytes. Tissue factor also circulates on membranous microparticles induced by inflammatory cytokines, where it drives pathological thrombus formation and the spread of thrombi. The amplification phase begins at the site of injury, where the platelets adhere to the injury site by contacting collagen and von Willebrand factor. The platelets also contact thrombin-producing tissue factor-bearing cells from the initiation phase and become partially activated. These platelets are called collagen and thrombin-stimulated, or COAT platelets. During amplification, thrombin, in a direct handoff from the tissue factor-bearing cells, escapes antithrombin and binds nearby COAT platelets. This triggers release of factor VIII von Willebrand factor complex and factor V from the platelet alpha granules and cleaves and activates factor VIII to factor VIIIa and 5 to 5a. This also separates the 8 from the 8 von Willebrand factor complex and simultaneously activates 11 coming from plasma and platelet alpha granules to make 11a, or activated 11. 9a from tissue factor bearing cells binds 8a on the COAT platelets, and 10a from tissue factor bearing cells binds 5a on COAT platelets. Further, 11a binds COAT platelet membranes, where it activates 9 from plasma and platelets to form 9a. Here again is the diagram of the initiation phase, just for your reference. This illustrates the amplification phase described in text in a prior slide. Thrombin, in a direct handoff from the tissue factor-bearing cell, escapes antithrombin and binds nearby COAT platelets. This triggers release of factor VIII and von Willebrand factor complex and also factor V, both from platelet alpha granules, and cleaves and activates factor VIII and activates also factor 11 coming from the plasma or from alpha granules to form 11a. 9a from the tissue factor bearing cells binds 8a on the COAT platelet, and 10a binds 5a on the surface of the COAT platelet. Further, 11 binds 9 on the surface of the COAT platelet, where it activates the 9 from both plasma and platelets and from the tissue factor-bearing cell. Activated protein C controls this process, but only in healthy, uninjured tissue. The propagation phase occurs on the coat platelet, which becomes further activated by thrombin, collagen, and von Willebrand factor. The tenase and prothrombinase complexes now assemble on the fully activated platelet surface. Tissue factor 7A and the 10A5A are considered the extrinsic mechanism, whereas 9A and the 9A8A complex are intrinsic. 10A5A is known as the prothrombinase complex, 9A8A as the tenase complex. The pathways on the platelet surface produce high-volume thrombin, which cleaves fibrinogen to fibrin and accomplishes fibrin polymerization and stabilization. The propagation phase is illustrated here as it occurs on the coat platelet, which becomes further activated by thrombin, collagen, and von Willebrand factor. Tenase and prothrombinase complexes now assemble on the fully activated platelet surface. The pathways on the platelet surface produce high-volume thrombin, which cleaves fibrinogen to form fibrin and accomplishes fibrin polymerization, cross-linking, and stabilization. Cell localization is central to the cell-based coagulation model. Tissue factor must be located on a cell that supports prothrombinase activity in association with platelets. Malignant cells that make tissue factor but don't support prothrombinase activity are not as effective at inducing clot formation. Platelet cell localization is essential. There's no tissue factor on platelets, so they must adjoin tissue factor-bearing cells. Also, there's no factor VII receptor on platelets. Thrombin cleaves the protease-activated receptor PAR to partially activate platelets. Coat activation moves phosphatidylserine to the outer leaflet to support tenase and prothrombinase. The coat platelets adhere to injury sites via a collagen receptor, glycoprotein 6. Meanwhile, glycoprotein 1b59 binds von Willebrand factor, further assisting coat platelet adherence to injury sites. Glycoprotein 2b3a binds fibrinogen and von Willebrand factor to support aggregation. The platelet provides surface receptors for factor 8, 5, 9, 10, and 11. Subsequent non-coat platelet layers dampen the reaction. Factor 12, high molecular weight kininogen, and prekallikrein make up the contact activation system. Contact activation is an in vitro phenomenon in which factor 12 is activated in the presence of a negatively charged surface, such as silica particles, kaolin, sealite, or ellagic acid. In vivo activation is induced by neutrophil extracellular traps, or nets. Stabilized by high molecular weight kininogen, activated 12 cleaves and activates factor 11, and coagulation proceeds as described previously. In the process, prekallikrein becomes cleaved, and the serine protease kallikrein bypasses factor 11 and cleaves factor 9. Kallikrein also triggers the inflammatory kinin system, creating a link with inflammation. Although contact activation is predominantly an in vitro phenomenon, the negatively charged surfaces of prostheses, such as mechanical heart valves, certain stents, and left atrial appendage implants, may trigger an in vivo process. Coagulation control via protein C is also a cell-bound activity. Thrombin binds constitutive endothelial cell thrombomodulin and activates protein C to form a serine protease. Free plasma protein S binds and stabilizes activated protein C, while bound protein S exerts less activated protein C stabilizing activity. Endothelial cell protein C receptor binds activated protein C to its surface, and the bound APC digests activated 5 and 8. Via this process, activated protein C activity is localized to healthy vascular beds. Meanwhile, the thrombin-thrombomodulin complex activates the thrombin-activatable fibrinolysis inhibitor. Thrombin binds endothelial cell thrombomodulin and activates protein C to form APC, or activated protein C, a serine protease. Free protein S binds and stabilizes APC, while C4b binding protein binds 60% of plasma protein S and lowers its protein C stabilizing activity. Endothelial protein C receptor binds APC to the surface, and the bound APC digests activated 5 and activated 8. Meanwhile, the thrombin-thrombomodulin complex activates the thrombin-activatable fibrinolysis inhibitor. The virtues of the former plasma-based system are listed here. The system models coagulation as a series of amplifying proteolytic reactions in which each protease cleaves and activates the subsequent substrate zymogen in the series. The system recognizes the participation of platelet phosphatidylserine as the essential assembly site and identifies the protime and partial thromboplastin time as corresponding to the extrinsic and the intrinsic systems. For the many years in which the plasma system held sway, these questions went unanswered. If there's a separate tissue factor pathway, why doesn't the 7A tissue factor activate enough factor 10 to compensate for a lack of factor 8 or 9 in hemophilia? If 8 or 9 deficiency both cause severe bleeding, why is 11 deficiency bleeding mild and variable? Why is little or no fibrin generated when the platelet count is less than 10,000, and why does aspirin reduce thrombin formation? The cellular coagulation system provides the answers. If there's a separate tissue factor pathway, why factor 10 activation by the 7A tissue factor complex doesn't compensate for a lack of factor 8 or factor 9 in hemophiliacs? Because the 7A tissue factor activation of 10 occurs on the wrong cell, the fibroblast or smooth muscle cell. Free 10 is inhibited by antithrombin and tissue factor pathway inhibitor. It can only work when it diffuses to a platelet if the platelet is nearby. If factor 8 or factor 9 deficiency both cause severe bleeding, why is 11 deficiency bleeding variable? It's because free 9 can transfer to the platelet as antithrombin has less effect, and free 9 on the platelet bypasses the need for factor 11. Finally, the cell-based system illustrates the importance of the platelet and how thrombocytopenia or aspirin suppression reduce thrombin production. Coagulation also contributes to wound healing. Fibrin attaches to platelet actin, a contractile protein, and the clot retracts. Macrophages secrete vascular endothelial cell growth factor and trigger neovascularization, and platelets secrete platelet-derived growth factor that attracts and stimulates fibroblasts and smooth muscle cells. A number of endothelial cell stresses generate adhesion molecules, adhesion molecule binding integrins, and cytokines, many of which participate in coagulation, thus tying inflammation closely to coagulation. We are learning that inflammation is a key to thrombosis, and the management of inflammation is antithrombotic. If thrombosis is hemostasis that occurs on cells, therapy could target the vulnerable cells such as endothelial cells. Also, aspirin suppression and thrombocytopenia of platelets slows coagulation, and statins' anti-inflammatory properties reduce recurrent venous thromboembolic disease by as much as 30 to 50%. We've completed this module in which we've looked at overlapping coagulation phases, initiation, amplification, and propagation, illustrating that all occur on cell membranes. Thank you for listening.